Hello everyone, welcome back. We are continuing with lipid metabolism, and in today's class we will be discussing the biosynthesis of triacylglycerol, phosphoglycerides and sphingolipids. So, these are the concepts that we will be covering. We will be covering the entire pathway where triacylglycerol biosynthesis is done, how the entire process is regulated, and we will also be looking at the overview of phospholipid synthesis, plasmalogen synthesis as well as sphingolipid synthesis. Okay. So, to start with, what is triacylglycerol? Now, this is the structure of glycerol. What is glycerol? It is 1, 2, 3 propane triol. Okay, this is glycerol. Now, we can attach fatty acid molecule to each and every. We can replace this OH group, I mean simply replace this hydrogen and we can replace it with fatty acid. Then we will get this molecule where each and every fatty acid molecule is referred to as acyl group ACYL. So, one acyl group, if this whole thing remains, and one OCOR1 COOR1 R1 is replaced, it is known as MAG or monoacyl glycerol. When this molecule or this H is replaced by another OCOOR2, another acyl group, it becomes diacyl glycerol. Simply when all the three, this is, as you all know from your chemistry lessons, organic chemistry, this is all known as SN1 position, this is SN2 and this is SN3, right, SN. So, when all the three positions are replaced by acyl groups, it is triacyl glycerol. So, this is the structure of triacyl glycerol, which is also abbreviated as TAG, all right. So, let us look how the whole molecule is synthesized. Now, you have some basic idea, right. We discussed during fructose metabolism as well as during glycolysis that these intermediates can be channeled towards synthesis of multiple lipids. So, this is the area where we are continuing from carbohydrate metabolism to lipid metabolism. Now, see the beauty of it, the precursors being glycerol, 3-phosphate and fatty acyl coenzyme A. How do we get fatty acyl coenzyme A? Simple, a fatty acid when it enters the fatty acid can get attached to the coenzyme A and it becomes fatty acyl coenzyme A. For example, when a 16 carbon palmitic acid is synthesized, it also can be represented as palmitoyl coenzyme A. Okay. The main intermediate that is or precursor that is required is glycerol 3-phosphate. Now, glycerol 3-phosphate can be obtained from dihydroxyacetone phosphate or DHAP by the action of an enzyme glycerol 3-phosphate dehydrogenase. So, glycerol 3-phosphate dehydrogenase is acting upon DHAP to form glycerol 3-phosphate. Who is helping or who is the reducing equivalent over here? NADH. So, NADH is required to donate one H plus into the reaction. Then what happens? The dihydroxyacetone phosphate, this keto group is reduced and it is replaced by an OH group, and this is thus becomes glycerol phosphate. Okay. So, simple glycerol, okay, this one glycerol, this OH has been replaced by one phosphate group. So, this is glycerol phosphate, and since it is the third position, this is 1, 2, 3, this is glycerol 3-phosphate. Okay. Why it is capital L? It is because of the configuration of this carbon, the second carbon, where if H is located on the right side, it is capital L, and if H is located on the left side, it is capital D, or in the opposite side, if OH is located on the left side, this is L, and if OH would have been located in the right side or this side, it was capital D. So, this is a type of isomerism. We are not going to details of chemistry right over here, but this is why it is known as capital L glycerol 3-phosphate. Now, this glycerol 3-phosphate can also be achieved from one intermediate or from one compound, that is glycerol. Very simple. We can attach one phosphate group directly to glycerol. Who attaches phosphate group? Kinase group of enzyme. Glucose, glucose 6 phosphate. Fructose, fructose 1 phosphate. So glucokinase, fructokinase. So here glycerol kinase. So glycerol kinase by acting on glycerol can lead to formation of glycerol 3 phosphate. Now what happens with this glycerol 3 phosphate? To this glycerol 3 phosphate, this H hydrogen atoms of this OH group will be simply replaced by acyl group from fatty acid. What is the enzyme? Simple acyl transferase. And if you wonder how fatty acyl CoA are being synthesized from fatty acid by the action of acyl coenzyme A synthetase, we all know that acyl coenzyme A with the, this is an ATP dependent mechanism which uses 2 ATP, that is known as activation of fatty acid. We all read that during beta oxidation of fatty acid. So that concept is already clear by now. So using that concept we can extrapolate our knowledge to find out that 2 acyl transferase are required where the 2 hydrogen atoms will be replaced. Now what is this? This is diacylglycerol. There are 2 acyl molecules, R1 or R2. Diacylglycerol, and in the third place 1 phosphate is added. So this is diacylglycerol 3 phosphate, which is also commonly known as phosphatidic acid or phosphatidate. Alright. So up to here, if it is clear, we can now go to the next slide. Now a bit of information about phosphatidic acid. This phosphatidic acid is actually present in a very small amount inside the cell, but it is a key molecule. It plays a sheet anchor role in all type of lipid biosynthesis. It is a key intermediate that can be converted to any type of triacylglycerol or glycerophospholipid, okay, any type of glycerophospholipid or TAG. Now let us see how it happens. So phosphatidic acid, so we need to get rid of this phosphate group somehow. How can we do that? We add phosphate with the help of kinase, glucose to glucose 6 phosphate hexokinase. If we were to do the reverse, we need phosphatase enzyme. Same concept also applies over here. So phosphatidic acid is acted upon by a phosphatase enzyme that get rid of this phosphate group. It is gone. Now we are left with 1,2-diacylglycerol. Simple. Because our goal is to reach to triacylglycerol, and as long as the phosphate group is here in the third position, we cannot attach a acyl group. So first we need to get rid of this phosphate group. It is done with the help of this enzyme phosphatase. The diacylglycerol is now ready to accept another acyl group in the third position, and with the help of another acyl transferase reaction, we are now getting the final desired product, that is triacylglycerol, which has got an acyl 3 different acyl groups R1, R2, R3. It can be same same type of acyl group may be present, but in whole thing generally it is known as triacylglycerol, where different side chains may be present in the acyl groups, right. So how this whole thing is regulated? You know, triacylglycerol synthesis is an anabolic process. All anabolic processes are helped by, you got it right, insulin. So whenever we are having increased amount of insulin, that is we are having a process where there is more and more fatty acid synthesis, that also helps in, ultimately that helps in synthesis of triacylglycerol, because triacylglycerol need fatty acid to start with. Fatty acyl CoA, if it is more and more, triacylglycerol will also be more and more. Who helps in synthesis of fatty acid? Increased amount of acetyl coenzyme A, right, and we all know that whenever we are talking about an anabolic reaction, insulin is stimulating both the synthesis of acetyl coenzyme A as well as synthesis of fatty acid. In turn it becomes fatty acyl CoA, and in turn it becomes triacylglycerol. Now when will this process will be hampered? Now you should know that where from we are getting acetyl CoA, we can get acetyl CoA from multiple sources, okay. We can get it from our diet, glucose, pyruvate to acetyl CoA, as well as from multiple non-carbohydrate sources that can be converted via glucogenic amino acid to acetyl coenzyme A, that is the process of gluconeogenesis. Now this whole thing is jeopardized in diabetes mellitus, where insulin is absolutely low or insulin is not acting. In that case what happens? This acetyl coenzyme A cannot be directed towards fatty acid synthesis, it cannot be utilized, and ultimately everything is diverted towards ketone body synthesis, that we all know. So, this is how TEG synthesis is regulated, that is by action of insulin, and this whole process is jeopardized in case of uncontrolled diabetes mellitus, where the phenomena of ketosis is happening, and we have already discussed in detail what are the features of ketosis. So, basically this is the triacylglycerol cycle, where upon requirement the triacylglycerol can be broken down to simpler compound like glycerol and fatty acid, okay, and this fatty acid is acting as a fuel, it goes outside the tissue, again it can be acted upon by glycerol 3 phosphate when the body is in anabolic mode, and again triacylglycerol will be synthesized, again it can by the action of lipoprotein lipase glycerol can be broken down. So, this whole thing occurs in tandem, where the triacylglycerol molecules are broken down and resynthesized during starvation. So, during starvation, when the body needs more and more sources of energy, the molecule is broken down, it is utilized, and upon refeeding the whole thing can be resumed, right. So, this was all about triacylglycerol synthesis and the regulation of triacylglycerol synthesis. So, remind it, in the triacylglycerol cycle the adipose tissue, blood and the liver, everything is involved. Now, we are moving on to phospholipids. So, what are phospholipids? Phospholipids are actually, as you have guessed, combination of lipid molecule with a phosphate group. What is so special about it? It is a amphipathic substance, mean it has got both hydrophilic as well as a hydrophobic region. Phospholipids are an integral part of membrane, okay, you already know that. But one thing to remember is the phospholipid synthesis, and few example of phospholipids are phosphatidylserine, phosphatidylcholine, ethanolamine and sphingomyelin. Now, where the phospholipid biosynthesis is happening, it is happening mainly in the smooth endoplasmic reticulum, that is SER, and the mitochondrial inner membrane, okay, inside the cell in eukaryotes. Mainly these are the two areas where phospholipid synthesis is happening. Now, again phospholipid synthesis, I told you when we need phospholipid or triacylglycerol, the key or the central molecule is phosphatidic acid. So, how phosphatidic acid, what is phosphatidic acid? 1,2-diacylglycerol 3 phosphate. How phosphatidic acid can be synthesized? Just before a couple of slides we saw it can be synthesized from glycerol 3 phosphate, where there is already a presence of phosphate group, and simply the two OH molecules are replaced by 2 acyl group by the action of acyl transferase. Well, we also saw a reaction where phosphatidic acid was acted upon by a phosphatase to form diacylglycerol or 1,2-diacylglycerol. Well, whenever there is a phosphatase enzyme, somewhere depending on the metabolic requirement, this reaction can be proceeded in opposite reaction with the help of a kinase. That is what exactly happens here. So, there is another route by which, depending on the requirement, phosphatidic acid can be synthesized from diacylglycerol with the help of the enzyme diacylglycerol kinase. Whenever there is a kinase enzyme, we need someone to donate the phosphate group, and in this case it is adenosine triphosphate. So, ATP is donating the phosphate group, one ATP is being hydrolyzed over, it is getting converted to ADP, and with the help of kinase, in the third place there is formation and entry of a phosphate group by replacing this hydrogen. So, 1,2-diacylglycerol is converted to phosphatidic acid. So, either we are replacing the OH group and phosphate group is already present, we are replacing with acyl group, or acyl groups are present and we are inserting the phosphate group. In any way we have to get, that is phosphatidic acid. So, this phosphatidic acid or this phosphate group is actually the hydrophilic part, okay, and the hydrophobic part is actually attached to it. Okay. How? This is known as head group attachment. Okay. Now, see this hydrophilic head, these are the hydrophobic part, the acyl groups, the arms. Okay. So, these are the ones that are represented here. So, now we need attachment of the head group, that is the hydrophilic head. How it happens? It happens by a phenomena where the phosphoric acid, when it condenses, it eliminates 2 molecule of water. So, the phospholipid head group is attached to a diacylglycerol by a phosphodiester bond. What is a phosphodiester bond? It is the bond which is formed, this is the phosphodiester bond OPO from both sides when a phosphoric acid condenses with 2 alcohol. So, over here there is one OH group that is attached to the head group, and there is an alcohol that is attached to the phosphatidic acid, that is an OH group. When a phosphoric acid condenses over here, 2 water molecules from here, HOH will be eliminated from here and one HOH will be eliminated from here. Ultimately, this will form a phosphodiester bond, and this is the basic structure of a glycerophospholipid. Okay. Now, when we look at the synthesis of a phospholipid, we will find that in most cases the head group is donated with the help of in CDP form, that is cytidine nucleotide. What is a nucleotide? It is a nitrogenous base, pentose sugar and a phosphate group. Okay. ATP, ADP, CDP, these are UDP, these are all nucleotides, where a nitrogenous base, that is cytidine, pentose sugar and a phosphate group is attached. When the phosphate group is not attached, it is known as nucleoside. We will be discussing them later when we are discussing about nucleotide metabolism, but this is the basic chemistry that we should be aware about, nucleotides and nucleosides. So, Eugene Kennedy found out that whenever phospholipid synthesis are taking place, the head groups are donated in the form, or the head group formation is taking place whenever there is a attachment of CDP or cytidine nucleotide. So, you can see over here, whenever there is phospholipid biosynthesis, the CDP supplies the phosphate group. Okay. So, here the phosphate group can be supplied via 2 ways. Number 1, as I told you, CDP means cytosine nitrogenous base, pentose sugar in the presence of in the form of ribose and a phosphate group. So, CDP can be in the form of diacylglycerol. So, the phosphate group can come from the glycerol side, or it can be in the form of a CDP alcohol. So, over here glycerol, so this the CDP diacylglycerol is getting attached to another alcohol, right, and over here a CDP alcohol is getting attached to 1,2-diacylglycerol. Anyway, in either of the mechanism, one molecule of CMP will be lost, I mean will be gone, and ultimately we are getting the formation of the basic glycerophospholipid structure, right. This thing is theoretical. Whenever a phosphoric acid condenses, it loses 2 molecule of water. This was thought earlier, but it is Eugene Kennedy who discovered this is not how it happens. It actually happens when alcohol specifically during head group donation is in the CDP form, and especially during condensation of the phosphate group. So, CDP phosphate either with glycerol or CDP phosphate with alcohol. So, these are the 2 strategies. Now, it has been found out that eukaryotic can use both the strategies to form the glycerophospholipid, whereas the bacteria can only use the first pathway, right. So, addition of polar group diacylglycerol phosphate with CDP, or polar group that is activated by CDP alcohol. So, now basically we can use the same concept where this phosphatidate, right, or phosphatidic acid can undergo combination with cytidine triphosphate. It undergoes hydrolysis and it forms CDP diacylglycerol, because CDP diacylglycerol can participate in all downstream reactions. So, over here what is happening? Phosphatidic acid is being converted to CDP diacylglycerol. This CDP diacylglycerol, to this now any head group can be attached using this principle. So, the head group can be serine, whether you see one CMP molecule is lost. So, one head group is coming in, one CMP molecule is lost and we are getting phosphatidyl serine. This structure can vary, but the basic, this head group can vary, but the basic structure will remain same. In this case what is getting attached? A glycerol 3-phosphate molecule, right. So, this is getting attached with the help of PG3 phosphate synthase. So, phosphatidyl 3 glycerophosphate synthase. Now, if we continue this with this molecule, that is phosphatidyl serine, it can be reacted upon by various enzymes to form various forms of phospholipids. You see, if it gets decarboxylated, it forms phosphatidyl ethanolamine. If it is methylated by the methyl donor, that is S-adenosyl methionine, it is also known as ADOMET, ADOMET, where by the action of methyl transferase it is converted to the form of S-adenosyl homocysteine, it is also known as ADOHCY. These are the abbreviations that means S-adenosyl methionine and S-adenosyl homocysteine. So, one addition of a methyl group, multiple methyl transferase, it leads to formation of phosphatidyl choline. Now, this phosphatidyl glycerol 3-phosphate, it can be acted upon by a phosphatase enzyme, where this phosphate group is first lost, it becomes an alcohol. To this alcohol another phosphatidyl glycerol 3-phosphate molecule can be added. So, this whole big structure is actually a mirror image of each other, and this is known as cardiolipin. It is a special form of phospholipid that is present in the heart. It has got multiple functions. We are not discussing the functional aspect of phospholipids over here, but we are discussing the biosynthetic pathway, but know this, cardiolipin is formed in this way. So, this is the summary. The basic concept is same. All alcohol can either be undergo a reaction with CTP to form the CDP counterpart, and thereafter they can act with diacylglycerol to form, in this case ethanolamine is forming phosphatidyl ethanolamine, choline can form CTP choline, it can form phosphatidyl choline. We also saw the interconversion of phosphatidyl choline to phosphatidyl ethanolamine to phosphatidyl choline by the action of methyl transferase. Now, these can also be interconverted by the action of phosphatidyl serine synthase. So, phosphatidyl serine synthase can convert phosphatidyl ethanolamine to phosphatidyl serine. Serine will come and ethanolamine will be gone. This is simple replacing of a head group, and this case also phosphatidyl choline and phosphatidyl serine can be interconverted by the help of interconversion of groups. Now, as I told you, bacteria can only utilize the first way. So, it can only undergo condensation with CDP alcohol, where a head group will come and it will form phosphatidyl serine in this case, all right. So, this is a summary depending on what are the two mechanisms by which phosphate can be donated using CDP, where multiple forms of phospholipids can be formed and interconverted among each other. Next, we will discuss plasmalogen. This is a complex structure, right. This might appear to be very intimidating, but we will dissect the whole thing very slowly. So, plasmalogens are a subclass of membrane glycerophospholipids that typically include, so basically the structure is the same, it is a phospholipid where there will be a head group, there will be a phosphate group, there will be two acyl groups, but these acyl groups vary in character, right. The first one is an vinyl ether bond. So, there is an unsaturation and there is an ether bond COC, right. So, this is the first position, and there is polyunsaturated fatty acid is present in the second position. Where the plasmalogens are used, they are highly abundant in neuronal, immune and cardiovascular cell membranes. Okay. So, let us see how plasmalogens are formed. Get your concept very right. The only goal over here is to displace the esterified fatty acyl group with a long chain alcohol. right. If we do that, we can see how the further process happens. So, initial process acyl transferase, everything is same, right. So, what happens, how can we get the long chain alcohol? So, what happens over here, you know, fatty acyl CoA and dihydroxyacetone phosphate condenses with the loss of a coenzyme A, and the first intermediate that is formed is 1-acyl dihydroxyacetone phosphate. Okay. So, with 1-acyl dihydroxyacetone phosphate over here, this is very basic reaction where basically the first carbon or the first hydrogen of dihydroxyacetone phosphate has been acylated, right. This we all understand. Now, in the second case, the second acyl group, it is treated in a different way. What happens in the second acyl group? It is reduced with the help of NADPH. When we are reducing acyl CoA, it is becoming a saturated alcohol because the coenzyme A is lost. Now, this whole the 1-acyl dihydroxyacetone phosphate and 1 saturated alcohol is acted together, it is condensed. The first RCO acyl group is lost and we are left with 1-alkyl dihydroxyacetone phosphate. Basically, what we have done, we have replaced the acyl group with 1 saturated or saturated alcohol. Okay. 1 alkyl group has replaced the first OH group of dihydroxyacetone phosphate. So, from 1-acyl dihydroxyacetone phosphate, it is 1-alkyl dihydroxyacetone phosphate. So, if we reduce it, what will happen? This dihydroxyacetone phosphate, I already told you, this ketone group will be reduced, and now we are getting the structure of glycerol, right. So, when this is reduced, this C double bond O will become CHOH. So, now what is the final structure, and we can also replace the acyl group. We already told you we can easily get rid of this phosphate group by the help of 1 phosphatase enzyme, and we can acylate 1 with the help of acyl transferase. So, ultimately this leads to 1-alkyl 2-acyl glycerol 3-phosphate. In this case, what is happening, we are not actually getting rid of the phosphate group, we are simply first replacing 1 acyl group in the second position, this is R3, right. So, this is 1-alkyl 2-acyl glycerol 3-phosphate. Okay. So, this part is clear. In the next part, what will happen? There will be head group attachment. Okay. So, head group attachment, the concept is absolutely same. So, this is basically in the form of CDP ethanolamine. So, in this case, if you are adding ethanolamine, CDP ethanolamine will be coming, and one CMP will be lost, and since this is a phosphate group, condensation will happen just like glycerophospholipid was being formed, and there will be formation of phosphodiester bond. So, there will be one structure where there is an saturated alcohol in the first, one polyunsaturated fatty acid in the second, and there is a head group in the third position. We are not there yet. What we need to do? We need to replace the saturated alcohol with an unsaturation. We need to induce double bonds. Who induces these double bonds? They are done by mixed function oxidase enzyme. So, one NADH is utilized, one oxygen molecule is incorporated, 2 molecules of water are lost, NAD is generated, and there is an unsaturation. So, this is the final structure of plasmalogen. Okay. And lastly, we are left with sphingolipids. So, what are sphingolipids? Sphingolipids are a class of lipids that contain a backbone of sphingoid bases, where the aliphatic amino alcohols is basically sphingosine. Okay. They were discovered in the brain extracts in the late 19th century, and were named after the mythological structure sphinx. Okay. So, that is why they are known as sphingolipids. This is the basic structure of sphingolipid, where the R can be an hydrogen group, then it will be known as ceramide. The R can be a phosphate group, then it will become ceramide 1 phosphate. The R can be a phosphocholine, it will be sphingomyelin. R can be sugar, it can be glycosphingolipid, irrespective of the structure. So, we will not be going into structural detail, we will be just seeing the overview of the important enzymes and the inhibitors that are involved in sphingolipid biosynthesis. For MCQ purpose, the most important is the starting material, that is L-serine and palmitoyl coenzyme A, where we get palmitoyl coenzyme A from palmitic acid is synthesized, one acyl it can undergo one coenzyme A transformation, where one coenzyme A will be coming and one SH group will be lost, and that will lead to formation of palmitoyl CoA. So, palmitoyl CoA and serine reacts to form 3-ketosphingolipid, right. That 3-ketosphingolipid is then reduced to form sphingolipid, okay, the enzyme being 3-keto reductase. The most important enzyme that comes later, I mean after this step, this known as sphingosine N-acyl transferase, also known as ceramide synthase. It leads to formation of dihydroceramide. Mind it, when we are talking about ceramide, it means the R group is simply a hydrogen molecule. So, from the basic structure we can derive multiple structures. So, if we replace the H, we can get multiple complicated version of sphingolipid. So, this dihydroceramide is then desaturated to form ceramide, and then subsequently with the help of multiple enzyme sphingomyelin synthase we can get sphingomyelin, where this H will be replaced by phosphocholine, alright. We can get galactosyl ceramide, while galactose will be replaced, we can replace it with the help of multiple sphingosine alcohol, that will sphingolipids, then that will be known as sphingosine. Next, very important, a glucose molecule can replace the H, that will be known as glucosyl ceramide. Now, what is most important in this whole slide? Most important are the inhibitors, because they are the ones that are targeted in case of multiple disease therapy. They are the molecules that are our molecules of interest in multiple research grade enzyme, for research grade enzyme inhibition, they being myriocin that inhibits the enzyme serine palmitoyl transferase, and fumonicin B1 or FB1, they inhibit the enzyme ceramide synthetase, and a very big name PDMP, that is 1-phenyl 2-decanoyl amino 3-morpholino 1-propanol. You may not remember this whole big name, but this PDMP inhibits synthesis of glucosyl ceramide. Now, one thing to note is the first few steps before the addition of head groups for all these takes place in the endoplasmic reticulum, whereas the final steps, that is the head group attachment, takes place in the Golgi complex. Okay. So, this was about the biosynthesis of triacylglycerol, phospholipid, plasmalogen as well as sphingolipid biosynthesis. These are the reference slides, and I thank you for your patient hearing.